Welcome to our deep dive on the Marburg virus. Now, for medical microbiologists, this isn't just another pathogen, it's a profound test of our entire public health system. And in this explainer, we're going to break down exactly why.
This single sentence, it just gets right to the heart of the Marburg challenge. It highlights what we call the therapeutic void, the complete and utter absence of a licensed cure or vaccine. You know, when you have no specific weapon against a virus, the system of response becomes everything. Every single protocol is a crucial line of defense. Oh, and just a quick note before we jump in, this entire briefing was put together using a Gemini deep search. So if you want to go check out the primary research for yourself, all the sources are linked up in the Notebook LM document.
All right, so to really get a handle on the challenge, we first need to understand the adversary. Let's break down the virology of Marburg.
Taxonomically, Marburg virus sits in the order Mononeogavirales, and, this is the important part, the family Filoviridae. Now that, of course, places it in the same notorious category as Ebola virus, which immediately gives you a sense of its pathogenic profile.
Now, within the species Marburg virus, there's a really critical fact that can get overlooked. See, it's not a single entity. It's actually two distinct viruses, Marburg virus, or MARV, and RAV virus, RAVV, and the genetic divergence between them can be as high as 21%. That number has massive clinical implications.
That 21% difference, it's not just some taxonomic detail. It's a direct threat in the diagnostic lab. A divergence that big can cause primer and probe mismatches in your molecular assays. What this means, and this is probably the single most important takeaway for diagnostics, is that any RT-PCR test has to be validated for both lineages. If it's not, you risk a catastrophic false negative.
So structurally, the Marburg virion uses a pretty common but very effective architecture. It's got a classic helical nucleocapsid that holds its RNA genome, and that's all enclosed within a lipid envelope. And that envelope, it's basically stolen directly from the host cell membrane as the new virus particles bud off.
And this is where we see its really unique morphology. Under an electron microscope, Marburg virus doesn't have one consistent shape. It's highly pleomorphic and famously filamentous. You'll see these long threads, but also the characteristic shepherd's crook, U-shaped forms, and even circles.
Okay, let's pivot to the disease itself, Marburg Virus Disease, and see how this builds from those initial symptoms into a really severe clinical picture.
The disease progresses with frightening speed. It starts very suddenly with these non-specific symptoms, like a high fever and a killer headache, but within just a week it can escalate to a severe hemorrhagic phase, and from day 9 on, patients often just spiral into shock and multi-organ failure.
This table gives us a much more detailed view that's super relevant for the lab because it links what the clinician is seeing to what we expect to find in the lab results. So in the initial phase, we see things like leukopenia and thrombocytopenia. This then quickly progresses to a really marked thrombocytopenia with elevated liver transaminases, before it all culminates in that late organ phase with severe coagulopathy, DIC, and signs of renal failure.
It's this rapid and severe progression that explains exactly why the Dutch response, as outlined by the RIVM and LCI, is so focused on procedure, not on prescription.
The guidelines are stark. They're definitive. As this quote from the LCI makes crystal clear, with no specific therapy available, everything, and I mean everything, falls back on the system. So let's take a look at how that system is designed to contain the threat.
The Dutch protocol is basically a rapid response cascade built on 3 pillars that you just can't compromise on. Step 1. A clinician with any suspicion at all must immediately consult a national VHT treatment center. Step 2. Because it's a Group A disease, even the mere suspicion triggers an immediate report to the Municipal Health Service, the GGD. And Step 3. All diagnostic samples are centralized and sent directly to the National Reference Lab at Erasmus MC.
And at the lab, this centralization means they have a whole range of tools ready to go. For that acute diagnosis, real-time RT-PCR is the absolute gold standard, since it detects viral RNA early in the infection. But the diagnostic toolkit also includes things like antigen capture, ELISA, for rapid screening during an outbreak, and serological tools like IgM and IgG ELISAs to diagnose later-stage disease or confirm a past infection.
Okay, so looking ahead a bit, this very absence of therapies forces us to think about the theoretical challenges we'd face down the road, especially when it comes to resistance.
We really have to differentiate between 2 types of resistance here. The first is intrinsic resistance, which, well, the virus already has.
It's incredibly good at evading our innate immune system by actively blocking the interferon signaling pathway. The second is theoretical acquired resistance. If we ever do develop an antiviral, let's say one that targets the L protein, we can pretty much predict that resistance would emerge through mutations in that very gene.
And what this all means is that right now, any antiviral susceptibility testing for Marburg is a preclinical, research-only activity. And while we have cell-based assays, the real gold standard for proving a drug's efficacy is the non-human primate model. This absolute reliance on NHP models, which can only be done in a handful of BSL-4 labs worldwide, creates this massive bottleneck in the drug development pipeline.
And that bottleneck leaves us with this one critical, forward-looking question for everyone in public health and medical microbiology. With drugs and vaccines still in development, our focus has to be on reinforcing the very system of detection, containment, and supportive care that right now stands as our only line of defense. And that concludes our briefing on Marburg virus. If you found this breakdown useful, definitely subscribe for more explainers on critical pathogens. We'd also really like to hear your perspective. What do you see as the biggest challenge in strengthening our systemic response? Let us know in the comments. Thanks for joining us.